बिस्मिल्लाहिर्रहमानिर्रहीम अस्सलामु अलैकुम।
Phosphorus is an important plant macronutrient making up about 0.2% of a plant's dry weight. It is a component of key molecules such as nucleic acids, phospholipids, and adenosine triphosphate, and consequently, plants cannot grow without a reliable supply of this nutrient. The uptake of phosphorus in plants occurs in ionic form. So you will see Pi in whole this lecture. So when there is written Pi, be clear that it is ionic phosphorus which is uptaken by plants. Ionic phosphorus is also involved in controlling key enzyme reactions and in the regulation of metabolic pathways. After nitrogen, phosphorus is the second most frequently limiting macronutrient for plant growth. A similar phosphorus. Phosphorus in soil, although the total amount of phosphorus in the soil may be high, it is often present in unavailable forms or in forms that are only available outside of the rhizosphere. Few unfertilized soils release phosphorus fast enough to support the high growth rates of crop plant species. In many agricultural systems in which the application of phosphorus to the soil is necessary to ensure plant productivity, the recovery of applied phosphorus by plants in growing season is very low because in the soil more than 80% of the phosphorus becomes immobile and unavailable for plant uptake because of absorption, precipitation, or conversion to the organic form. So be clear that the phosphorus applied to the soil get fixed and then released slowly in the ionic form, and plants uptake phosphorus in ionic form. Soil phosphorus is found in different pools such as organic and mineral phosphorus. It is important to emphasize that 20 to 80% of phosphorus in soil is found in the organic form, of which phytic acid is usually a major component. The remainder is found in the inorganic fraction containing 170 mineral forms of phosphorus. Soil microbes release immobile forms of phosphorus to the soil solution and are also responsible for the immobilization of phosphorus. The low availability of phosphorus in the bulk soil limits plant uptake. More soluble minerals such as potassium move through the soil via bulk flow and diffusion, but phosphorus is moved mainly by diffusion. Since the rate of diffusion of phosphorus is slow, high plant uptake rates create a zone around the root that is depleted of phosphorus. Plant root geometry and morphology are important for maximizing phosphorus uptake because root systems that have higher ratios of surface area to volume will more effectively explore a large volume of soil. In certain plant species, root clusters are formed in response to phosphorus limitations. These specialized roots exude high amount of organic acids which acidify the soil and chelate metal ions around the roots, resulting in the mobilization of phosphorus and some micronutrients. Phosphorus uptake across the plasma membrane. The uptake of phosphorus poses a problem for plants since the concentration of this mineral in the soil solution is low, but plant requirements are high. The form of phosphorus most readily accessed by plants is ionic phosphorus, the concentration of which rarely exceeds 10 mm in soil solution. Therefore, plants must have specialized transporters at the root-soil interface for extraction of ionic phosphorus from solutions of micromolar concentrations as well as other mechanisms of transporting ionic phosphorus across membranes between intracellular compartments, where the concentration of ionic phosphorus may be thousandfold higher than in the external solution. There must be afflex systems that play a role in the redistribution of this precious resource when soil phosphorus is no longer available or adequate. You will see two different terms, afflux and influx. Influx is the movement from outside to the plasma membrane, and afflux is movement of ions from plasma membrane to outside. The form in which ionic phosphorus exists in the solution changes according to pH. The pH for the dissociation of H3PO4 into H2PO4 and then into HPO4 are 2.1 and 7.2 respectively. Therefore, below pH 6, most ionic phosphorus will be present as the monovalent species, whereas H3PO4 and H2PO4 will be present only in minor proportions. Most studies on the pH dependence of ionic phosphorus uptake in higher plants have found that uptake rates are highest between pH 5 and 6, where H2PO4 dominates, which suggests that ionic phosphorus is taken up as the monovalent form. Ionic phosphorus does not enter simply as H2PO4 or HPO4, both of which would lead to membrane hyperpolarization. It is likely that ionic phosphorus is co-transported with positively charged ions. Co-transport of ionic phosphorus with a cation involving a stoichiometry of more than one carbon or more than two carbon would result in a net influx of positive change and hence lead to the observed membrane depolarization. The cytoplasmic acidification associated with ionic phosphorus transport would suggest that the cation is H1, but acidification would occur regardless of the nature of the cations if the transported species were H2PO4, since it would undergo a pH dependent dissociation in the cytoplasm or to H2, HPO4 and hydrogen. Compartmentation of phosphorus. Maintenance of stable cytoplasmic ionic phosphorus concentration is essential for many enzyme reactions. This homeostasis is achieved by a combination of membrane transport and exchange between various intercellular pools of phosphorus. These pools can be classified in a number of different ways. First, according to their location in physical compartments such as the cytoplasm, vacuole, apoplast, and nucleus. The pH of these compartments will determine the form of ionic phosphorus. The second pH for H3PO4 is 7.2, so ionic phosphorus in the cytoplasm will be approximately equally partitioned between the ionic forms, whereas in the more acidic vacuole and apoplast, H2PO4 will be the dominant species. Third, by the chemical form of phosphorus, such as ionic phosphorus, phosphoric esters, phosphoric lipids, and nucleic acids. The proportion of the total phosphorus in each chemical form, except phosphorus in DNA, changes with tissue type and age and in response to phosphorus nutrition. Third, according to the physiological function as metabolic stored and cycling forms. Regulation of ionic phosphorus uptake. When the supply of ionic phosphorus is limited, plants grow more roots, increase the rate of uptake by roots from the soil, retranslocate ionic phosphorus from older leaves, and deplete the vacuolar source of ionic phosphorus. Conversely, when plants have an adequate supply of ionic phosphorus and are absorbing it at rates that exceed demand, a number of processes act to prevent the accumulation of toxic ionic phosphorus concentrations. These processes include the conversion of ionic phosphorus into organic storage compounds, for example, phytic acid, A reduction in the ionic phosphorus uptake rate from the outside solution and ionic phosphorus loss by efflux, which can be between 8 and 70% of the influx. Any or all of these processes may be strategies for the maintenance of intracellular ionic phosphorus homeostasis. Phosphorus translocation in whole plant. In phosphorus sufficient plants, most of the ionic phosphorus absorbed by the roots is transported in the xylem to the younger leaves. Concentrations of ionic phosphorus in xylem range from 1 millimeter in phosphorus ionic phosphorus starved plants to 7 millimolar in plants grown in solutions containing 125 millimolar ionic phosphorus. There is also a significant translocation of ionic phosphorus in the phloem from older leaves to the growing shoots and from the shoots to the roots. In ionic phosphorus deficient plants, the restricted supply of ionic phosphorus to the shoots from the roots via xylem is supplemented by increased mobilization of stored phosphorus in the older leaves and retranslocation to both the younger leaves and growing roots. This process involves both the depletion of ionic phosphorus stores and the breakdown of organic phosphorus in the older leaves. In the xylem, phosphorus is transported almost solely as ionic phosphorus, whereas significant amounts of organic phosphorus are found in the phloem. Physiological functions of phosphorus. Part of all living cells. All living cells require a continuous supply of energy for all the processes which keep the organism alive. For example, adenosine triphosphate and phosphorus is a part of adenosine triphosphate. So, all living cells and all living things need energy, which is provided by ATP, and phosphorus is an integral part of ATP. Essential part of photosynthesis and respiration. Metabolic process involve a series of chemical interaction that gives NTPs, NADH and FADH2 as an energy source. All of these have phosphorus, so phosphorus is an important part of these processes. Here you can see that in photosynthesis and respiration, ATP either is required or emitted, so both processes are dependent on phosphorus. Genetic transfer. Phosphorus is a vital component of the substances that are building blocks of genes and chromosomes, so it is an essential part of the process of carrying the genetic code from one generation to the next, providing the blueprint for all aspects of plant growth and development. An adequate supply of phosphorus is essential to the development of new cells and to transfer of genetic code from one cell to another as new cells are formed. Nutrient transport. Plant cells can accumulate nutrients at much higher concentration than are present in the soil solution that surrounds them. This allows roots to extract nutrient from the soil solution where they are present in very low concentrations. Movement of nutrients within the plant depends largely upon transport through cell membranes, which requires energy to oppose the forces of osmosis. Here again, ATP and other high energy phosphorus compounds provide the needed role in signal transduction. Phosphorus has an important role in signal transduction. Inositol triphosphate together with DAG is a secondary messenger molecule used in signal transduction and lipid signaling in biological molecules. While DAG stays inside the membrane, IP3 is soluble and diffuses through the cell. It is made by hydrolysis of PIP2, a phospholipid that is located in the plasma membrane by P. Thank you very much.